Be talking about microbial growth. This is going to be chapter 4 in your textbook. Today's objectives are to describe the principles of microbial growth, both in nature and in laboratory conditions. We're going to be evaluating the environmental and nutritional factors that influence microbial growth, and we're going to examine the methods to detect and measure microbial growth. So first thing we're going to look at is sections 4.1 through 4.7. These are going to be the dynamics of microbial growth. So first, we want to know exactly what bacterial growth is. Well, this is going to be cells growing larger, but also splitting into other cells. So we're going to use a new term that you may not know yet. This is binary fission. Binary fission is similar to a topic you might have covered in general biology. If you remember cell division, this is going to be very similar. It's just going to be the bacterial version of it. So just like with cell division, cells can get longer or larger as their DNA replicates and they produce more proteins, enzymes, whatever, in order to survive. However, as a cell starts to get too large, it runs into some problems. So the main driving force of nutrients and other and waste to either bring stuff into the cell or out of it is diffusion. Well, if a cell gets too large, diffusion is no longer viable, and that can cause a large cell to die. So what our cells are going to do are actually divide as they get larger. So the DNA will be sequestered into either side as daughter cells will form. And then those cells are going to pinch off and divide into two cells, and they can separate or they can stay together, just depending on the bacterial morphology. So different strains are going to have a specific generation time. A generation time is just how long it takes for one cell to double its, or not cell, but one population to double its population size. So that's going to be the generation time. And we'll look at that more in the next slide. So let's look at the exponential growth of bacteria. So bacteria are going to follow what can be called an exponential growth curve for a time. Now, I do put exponential in quotations because many situations it's actually going to follow something called a logarithmic curve instead of an exponential curve, but we'll cover that later. So first thing we want to do is get comfortable with this formula. This formula is going to be the exponential growth formula. Now, there's a whole lot of letters in here, and it can be a little scary, but let's break it down. So first we're going to have NT, N sub T. Well, N sub T just tells us the total number in a population. Okay, so we're trying to solve for N sub T most of the time. So N sub T is the population, and that's equal to N sub 0, or N naught. And this is the initial population. And then that's equal to, and then you multiply that by 2 to the nth power. N is just the number of generations that have passed. So with this, we can take that generation time that we looked at and figure out how many generations have passed. So in this example, we have generations going up every 20 minutes. So this example bacteria has a generation time of 20 minutes. So every 20 minutes, it'll double in size. So what we're going to do is we're going to do an example. We're going to look for N sub T. Oops, that's not right. Here, let's erase all. And so we're going to do N sub T, and that's equal to N naught times 2 to the nth power. So just for argument's sake, let's go ahead and use 4. So our initial population size is going to be 10. All of these started with the same population, we're just tracking it over different times. So 10 times 2 to the 4th power, because we're using this 4th generation. So like you know, or if you may not, but just to review some of your math, if you're raising something to the 4th power, you're just multiplying it by itself 4 times. So we have 2 times 2, that's 4. 4 times 2, that's 8. 8 times 2 is 16. So let's go ahead and simplify to 10 times 16. Okay, and then you just multiply 10 times 16 to get 160. And we can compare that to our graph and see, yes, there are 160 cells at the end of 4 generations. So this is pretty simple, and it can just be a little scary at the beginning. Okay, so let's look at biofilms, because biofilms are going to be a very interesting thing when you're looking at bacterial growth. So biofilms are going to be an extra polymeric substance. So this just means it's substances that go outside of the cell, and it'll actually give it somewhere to live. So this is going to be a slimy substance that coats drains, rocks, or even your teeth. Yeah, so if you've ever gone to the beach and you feel this, or if you go to a river and feel a rock and it feels extra slimy, there could be a biofilm on there. If you haven't washed or cleaned your bathtub in a while, there could be a slime on the drain, and that's going to be a biofilm. And if you don't brush your teeth frequently enough, that plaque is just biofilm giving the bacteria somewhere to adhere to. And this is going to be made of polysaccharides. So polysaccharides are usually going to be, if they're outside of the cell, are just going to be an area to collect water and for cells to grow on. So biofilms are going to be something that you need to be aware of as a healthcare professional, because biofilms can grow on different surfaces. So like a catheter or other medically inserted devices can all grow biofilms. So you have to make sure that they are sterile and handled in an aseptic setting. Okay, so how do biofilms develop? So basically you have these bacteria that are going to move to a surface, and they're going to adhere to it and start to produce that extracellular polymeric substance, or EPS for short. Now this substance is going to continue to grow, and other bacteria can actually latch on. So this one bacteria, well, it's going to make a home where other things can come in and grow too. And so the cells can communicate and create channels to channel that waste and nutrients to pass in. And eventually some of those cells can detach and go off to make new biofilms. Okay, so now let's look at bacteria in a laboratory setting. So in nature, you can have a bunch of bacteria found together. And when you're working in a lab, you typically don't want to use multiple types of bacteria together unless there's a specific reason. So what we're going to do in a lab is isolate a single type of bacteria, and that single type of bacteria can produce a pure culture. So a pure culture can have millions of bacteria of the same type, and they all originated from a single cell. So you might see on here there's some bacteria growing together, like some red and white, but what we're looking for are pure cultures like this white one. Or if there's one that's just pure red, I'm not sure if I see it on here because it's kind of taken in a fuzzy style. But those are going to be your pure cultures. So with those, remember there are millions of bacteria that all started from a single cell, and it's a single type of bacteria. Those are going to be two of the biggest points when you're looking at pure cultures. So how do we get pure cultures? Well, we're going to use something called the streak plate method. So I'm going to draw a little petri dish up here, or agar plate, however you say it. So what we're going to do is we're going to take this mixed culture. So you're going to have multiple types of bacteria in this culture, and you're going to take a inoculating loop. So if you have a wire loop, you will flame it in order to sterilize it. If you're using plastic pre-sterilized loops, you'll just grab a new one every time. So you'll take your sterilized loop, you're going to inoculate it, dip it into the bacteria, and then you're going to streak this first area, and that's going to look kind of like this. Just go back and forth with it. Okay, and then if you're using plastic, you would discard that loop and grab a fresh sterile plastic loop. If you're using a wire loop, you would flame it again until it gets red hot, and then you let it cool down naturally. You don't wave the loop through the air, and you don't blow on the loop, because both of those ways can get more bacteria on the loop. So then you'll take that loop, and you need to make sure it cools first if you're flaming it, because if it's not cool, it'll just burn whatever it touches. Not a good idea. So You're going to take that loop and streak through the starting, or the first one, but you're going to go past. So it'll look kind of like this. And what that should do is it should drag some of the bacteria from that first one, but it won't have nearly as much. And then you'll get a new loop if you're using a plastic pre-sterilized one, or you'll flame the inoculating loop again, and then you'll streak through it one more time. And that's going to give us this pattern that we can see over here. So we have our first streak right here, our second streak right here, and our third streak that goes over here. And that third streak is the one that'll actually give you isolated colonies. Now, in your lab section of this class, you'll be doing this during week 3. So, and I do believe that you use plastic pre-sterilized loops. So just make sure that every time you do it, you get a fresh loop, and you only dip it once for the first streak, and then you don't dip it again. Okay, so let's look at how the bacteria will grow in lab conditions. A lab condition, growing the bacteria in a growth medium, like on an agar plate or in a nutrient broth, this is going to be considered a closed system because nothing is going into the system and nothing's coming out. So there's no new food going in and none of the waste is going to be taken out, and this will create a growth curve we see in the lab. So first we have our lag phase. Now this happens because with exponential growth, it happens very slowly at the beginning. So if we consider our formula from earlier where we have 10 times 2 to the first power, well then, because only one generation passed, it goes to 10 times 2 is 20. Well, that didn't grow very fast. But then once we start getting a little bit further, 10 times 2 to the fourth, that's going to be 160. So eventually it'll start to go much faster, but at the beginning, it doesn't grow very fast at all. So that's why we call it the lag phase, because there's growth, but it's fairly slow. Next is going to be the exponential phase. So the exponential phase is once that growth actually starts to demonstrate the large leaps that exponential formulas will show. We will call this first half of the curve, this is called a J curve because it kind of resembles a J, and this, or it's also called an exponential curve. So, but in a lab condition where we have a closed system, that won't last for long. Eventually, we're going to reach this this stationary phase. So the J curve is just from the beginning to the start of the stationary phase. And then we can once we get to the stationary phase, this is where the death of the bacteria is actually going at the same rate as the growth of the bacteria. So that's kind of like the carrying capacity of the system, and eventually conditions will change, but we'll look at that in a moment. So this can, from to the beginning of the death phase, from the beginning of the lag phase, this can be called an S curve because it has a vaguely S shape to it. Okay, so as the stationary phase is progressing, resources will start to dwindle. There will be less food, less necessary chemicals for the bacteria, and they're actually going to start to accumulate waste products, which can create a toxic environment. So the starvation and poisoning of the bacteria can actually lead to the death phase. And this is where you have a sharp drop in population size because of those conditions. And we'll cover those more in a moment. After the death phase, you'll see that it starts to taper off in the phase of prolonged decline. So what's going on here is you have some slight adaptations to be able to survive the conditions that are present. So these bacteria are actually going to be slightly different than what you had at the beginning because they've adapted to the problematic conditions. Okay, so I was talking about metabolites a little bit earlier, or the waste products. So let's look at what metabolites we have. First, we have our primary metabolites. Now let's first orient ourselves with this graph. This blue line is going to be your population, So blue is your bacterial pop. And then the solid pink line are your primary metabolites, and the dashed pink line are your secondary. So your primary metabolites are normal waste products like ammonia, carbon dioxide, etc., etc. So these ones will actually be produced at a similar rate to the population growth. And then they'll start to level off once you reach the secondary or the stationary phase. But secondary metabolites are going to be something very different. They're special. They don't really start to grow very fast until you reach that stationary phase. Now, what's happening here is a lot of the food in the system is going to be gone. So these different organisms are going to be preparing for starvation. So one type of secondary metabolite is going to be endospores, where bacteria are like, okay, this condition is not good. Let's put ourselves in suspended animation in an endospore. Another thing that they can produce are antibiotics. So bacteria, they're pretty crazy. When they fight over food or resources, they don't hold back. They resort to chemical warfare. So these bacteria try to create chemicals that will kill their enemies, but not themselves. So bacteria are wild. And so some can produce antibiotics in order to secure resources for themselves. And this all happens because there is not enough food. Okay, so now that we've gone over how they kind of grow in a laboratory and some of their strategies to preserve resources for themselves, let's talk about temperature requirements. So different organisms have an optimal temperature range where they can thrive in. So think about humans. We have what we say a normal range is 98.6 plus or minus a couple degrees. Now, once you get outside of that, you can start to have problems. Well, same thing with bacteria. Bacteria or any other organism will have changes in their protein activity depending on the difference in temperature. So different proteins will have an ideal temperature and acidity level. So if you have fluctuations in temperature, these proteins won't be as active, so growth can slow down. So what we usually look at are mesophiles or mesophiles. These are going to be what exist at normal temperatures like 30 to 40 degrees Celsius. On either side, they can drop off pretty quickly. And we utilize that pretty well with like refrigerators, which make it so these mesophiles don't grow very fast. However, we do have archaea bacteria, and those ones will typically exist at extreme ranges. So first we have, going down for mesophiles, we have psychrotrophs. These ones can live at lower temperatures. They're more similar to mesophiles with where they exist, but they can go down to freezing, but they don't do well past freezing. So these are going to be the kinds of archaea bacteria that could potentially grow in your fridge. And then we can have psychrophiles, and these ones are ones that actually like to be very cold. These are going to be ones that can grow in frozen conditions. And then above mesophiles, we have thermophiles. These ones like to live in hot water. So think about some of the geothermal pools in Yellowstone, for example. They like hot water, and they've developed a way to live in it. And then you have hyperthermophiles. These ones live at boiling water or even above that. These bacteria are pretty nuts. Typically, you're only going to find them deep in the ocean around a volcanic vent, underwater vent, where they live. Yeah, they're pretty wild. Okay, next we're going to look at oxygen requirements. So oxygen, growing up, you think oxygen is great for life. It's something that's necessary, and to a point it is true. But there is also truth to say that oxygen is actually kind of toxic. So if you've ever heard about drinking grape juice will give your body antioxidants, oftentimes that can just sound like a word. Well, what is an antioxidant? An antioxidant is going to be a substance that fights oxygen. So oxygen is actually pretty reactive. If you've ever had a bicycle that gets rained on and you notice the chain rusts, that's because oxygen is reacting with it. It's actually ripping electrons off the metal. Well, oxygen can also do that in your body or in bacteria. unless they have specific mechanisms to protect them. And that's what this bottom part talks about. So different organisms have different tolerance for oxygen, whether they've evolved or adapted to the presence of oxygen. So our first one that we're going to look at is an obligate aerobe. These ones need oxygen. So in a test tube, they're going to grow more towards the top. Yeah, and then they're going to have different mechanisms to fight oxygen. One of those that we're going to look at, and you're going to look at in the lab section, is catalase. The catalase enzyme can break down like hydrogen peroxide. A facultative anaerobe is going to be one that grows best with oxygen, so you'll see a large distribution at the top, but then it can also grow without oxygen at a slower rate, so you'll get some scattered at the bottom. And then you have obligate anaerobes. These ones are going to be the ones that cannot grow in the presence of oxygen, so you'll see a distinct shadow at the top where there's nothing growing, and then you'll have bacteria growing in the bottom. These ones do not have catalase, and that'll be important once you get to week 5 of your lab section, where you're trying to use the catalase test to identify whether something is an obligate anaerobe or an obligate aerobe, or facultative aerobe. And then you also have microaerophiles. These ones grow if there's small amounts of oxygen, so they're like Goldilocks. They don't like too much and they don't like too little. They like it just right. And then you have aerotolerant anaerobes. These ones grow equally well with or without oxygen, and they'll have a distribution throughout the test tube. Water requirements is the next topic. Water is needed for life. Full stop. So how do organisms maintain water? Well, they can do that by using dissolved substances. We call those solutes. Water would be the solvent. So just a basic recap on osmosis. Osmosis is water moving through a semi-permeable membrane, and it will follow the higher concentration of solutes. So if there's more solutes inside of a cell, the water will flow into a cell. If there's more solutes on the outside of the cell, the water will leave the cell. And this isn't just like a whole number, it'd be more like the concentration. If the concentration of solutes is greater on either side, the water will follow that way. So what would happen if you have something very sugary, like fruit preserves, like strawberry preserves? Well, you put strawberries into a large amount of sugar. And so what ends up happening is if any bacteria wants to grow on the sugar, it has a hard time growing unless it's tolerant to that sugary condition, because the sugar is actually going to cause water to leave the cell. And we can do the same thing with salt. Salt can sap the water out of organisms. And so that's why salted meats are able to stay good for a long amount of time. So some organisms, though, are able to live in salty conditions. These are typically archaea, and so they're able to keep their water in other ways. So here's just a quick breakdown of everything I've talked about. In addition, there's also pH, acidity. This one doesn't have a whole lot to go over. If they like to live in acidic conditions, so pH 5.5 or below, they'd be classified as acidophiles. If they like to grow in basic conditions, 8.5 or above, they would be alkalophiles. And then if they like to be between 5 and 8, they're neutrophiles. Okay, and then these organisms are going to have 7 required major elements. And so some of those are going to be carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus. Carbon, oxygen, and hydrogen are going to be those basic components of pretty much every organic molecule you can think of. They're the basic parts of sugar and lipids. You can have a complete lipid and complete sugar with only carbon, oxygen, and hydrogen. With nitrogen, amino acids and nucleic acids can also use nitrogen. Some amino acids like methionine or serine, I think, Don't quote me on that one. We'll have sulfur. I think it's cysteine, actually. Cysteine, not serine. Cysteine. And then phosphorus is going to be in nucleic acids, membrane lipids, and ATP. Well, where is it in membrane lipids? Well, sometimes you can have a phosphate head group, and that's just going to be a lipids or a fat with a phosphorus on the top. So we call that the phospholipid bilayer if there's two layers, and that's going to be the cell membrane. ATP, you might remember, is the energy currency of a cell. So T means tri, P means phosphate. Phosphates have phosphorus in them. And then you also have potassium, magnesium, calcium, and iron. These are just required for certain enzymes, and they just give certain functions. Okay, carbon and oxygen sources. Different organisms are going to acquire their energy and their carbon in different ways. Humans, for example, we like to get both in the same way, and that's by eating. So we would be what's called a chemo-organo-heterotroph. These are going to be organisms that obtain organic compounds like sugars, amino acids, lipids, etc. They're going to obtain these chemical substances that contain energy. So for this, that would be that chemo. And because it's organic, we get organo. And then we also will get the carbon from the same organic sources. So if it gets its carbon from something else, we would call that a heterotroph. A chemolithoautotroph. So chemo means that it's going to get its energy from chemicals, chemical reactions. Litho is going to mean that it's not organic. So these are going to get energy from other chemical reactions. And that can be doing things with ammonia, iron, or any of these other substances. And so one of the examples are going to be archaeobacteria that grow around volcanic vents down in the ocean. And their carbon source, though, is going to be carbon dioxide. So it's going to grab it from its environment, the carbon. So with that, that's what we call an autotroph, is if it gets carbon dioxide from the environment. Auto. And then we can have a photoheterotroph. It gets its energy. Photo means sun, or light. So it gets its energy from light, and it would use organic compounds. And so I'll think like a Venus flytrap. It gets its energy from sunlight, but it lives in nutrient-poor soil, so it needs to break down other organic compounds for carbon. Well, that might not be correct, but it gets maybe not. I don't know. Ignore the photoheterotroph. I don't have a good example for it. Photoautotroph, that one's going to be your classic plant where it gets its energy from sunlight and its carbon source is oxygen. So growth media. Growth media, we have two different types, but there's going to be more types later on, so take that with a grain of salt. These are like the largest classifications. This is the biggest umbrella. So we either have complex or chemically defined. So we'll start with complex. This is highly variable, and it can be mixed with other special ingredients. So all that basically means is we have general things that we throw in there like peptone and beef extract, but we don't actually really measure it very much. We just throw that in water and call it good. And that's a nutrient broth. It's complex because it's got a lot going on. Chemically defined is going to be something that's controlled, and we have specific chemicals. So an example of that is a glucose salts broth. This uses very specific chemicals. They're all listed here. And if this was lab instructions, they would also give you instructions for how to mix the correct concentration. So complex is if you just need something to grow, you can throw it in there. chemically defined is very specific, and you're planning to do something with that. And so what you could be planning to do with that could be a selective or a differential culture media. So selective allows for growth of specific organisms but restricts others. So think about if you were trying to grow an antibiotic-resistant bacteria, and so you put a little bit of ampicillin into your agar, and then that could reduce the growth of other organisms, but allow the growth of that ampicillin-resistant bacteria. So something that this happens quite often in demonstrations in college is when you're doing bacterial transformations, you'll actually give things like E. coli resistance to ampicillin, but then you'll kill it preferably before it gets out, so that you don't release problematic strains. A differential is going to be a culture type that will create a visible change or observable change. So for example, this this pink one shows that some of them turned pink and some of them did not. And so this just shows that some metabolic activity took place in one but not in the other. This will be important for your lab. In topic 5, you're going to be asked to identify an unknown bacteria by using tests. And so you're going to be using some differential growth medias once you get there. But more on that later. There will be more on that later, and your lab professor will go over that with you. Another example could be if you grow something on blood agar, you could see how much it breaks down those heme groups with beta hemolysis or alpha hemolysis breaking down more or less, and then possibly no hemolysis going on. So here's just an example of how these can be used together. So routine medium. So this could be your complex medium. It's not going to be, or it could be a complex, or it could be a controlled medium. It doesn't really matter. But just basically, this one's not being selective or differential. So any bacteria could grow, A, B, C, E, or D, or E. But on a selective medium, this one's selecting for specific traits. So A, C, and E can grow on this, but B and D did not. A differential medium will have an observable change based on some sort of activity in the cell. So A, C, and D are going to stay the original color, but B and E change color. But if you really wanted to mix that together and identify if you knew the traits of specific bacteria and were trying to look for it, you could mix both of these two together and get a selective and differential medium. This will prevent B and D from growing, but it'll allow E to grow. So then you would be able to narrow it down based on what you're doing. So these are just a few media used to grow bacteria. A few of the ones that you're really going to want to know are going to be like glucosol agar, McConkey agar, nutrient agar. These are going to be some of the ones that you're going to use in topic 5 of your lab class, where you're going to identify a bacteria. So you might want to look at these now, but definitely look at them once you get to that point in your lab. Okay, so enrichment cultures, these are going to be a type of selective culture. These ones are providing ideal conditions for a specific organism, but inhibit the other organisms. So it's going to help your target organism grow while preventing others. So you can take your medium that contains the specific amount of substance that can help your target organism grow, add a mixed culture, incubate it, and hopefully your culture would grow while the other ones would die. And so once you inoculate a plate and incubate it, then you're going to have your enriched sample. Okay, so now we know how to grow bacteria in a lab, and we know what their requirements are in nature as well. So let's look at how we are able to count them. So something that we're going to do is we're going to count specific cells via dilutions, and there's a few different ways we can do that. So dilutions, you're going to take some of the original volume and dilute it with pure water. That's just how it goes. And we'll look more into how dilutions work on the next slide, I believe. So a microscope count, what you're going to do is you're going to take your bacteria, put it on a slide with a counting grid, and then you're going to count it one by one. This is kind of work that whoever's in charge of a lab would not do, and they would outsource it to one of their grad students or undergrad students. It's not very fun, but it needs to be done for research. Another option is flow cytometry. This one's kind of special. What you're going to do is you're going to take a sample of cells, pass it through, and then they'll go single file. You're going to shine a laser through there. And based on the way that light works is it will diffract or scatter based on different factors. So size is one factor, and granularity, so how much stuff is going on in the cell. So you can calibrate your flow cytometry to count specific cells based on their traits. Okay, next are going to be variable cell counts. These are going to be plate counts and membrane filtration. First, we're going to start with plate counts. So with plate counts, what you're going to want to do is perform a serial dilution. So serial dilutions are done by taking your original culture and taking 1 milliliter, diluting it into 9 milliliters of water. Now this will give you a 1 to 10 dilution. Well, you're going to want to cover your bases, so you're going to do that a few more times. You'll take 1 milliliter of this concentration and add it to 9 milliliters of water, and then you'll get a 1 to 100 dilution factor. And you'll do it again and again. So this way, you're going to do it a few times just so then you can get usable results. Now, if you look at this example, these ones just give you way too many cells to count, way too many colonies. So we would put like a TNTC, too numerous to count, if we were to look at that. It's just not happening. On the other side, if you dilute it down to 1 in 10,000, well, now you don't quite have enough results. So that one's not good either. What you want is to have a good amount, like 30 to 300. And so each of those growths will indicate that there was probably one bacteria that was the founder of the colony. And so what you can do is take the count, multiply it by the dilution factor to find out the concentration of your initial sample. So 50 times 1,000 would give you 50,000. So if you counted 50 on the plate. Yeah. So you can either do this with a spread plate where you spread it, spread the broth on a solid agar, or you could pour it and do the pour plate method where you mix melted cooled agar, not quite solidifying but cooled, and mix that with your desired culture. Something cool about the agar is it will melt at a certain temperature, but you can bring it below that temperature without it solidifying again until it reaches another number. But pretty cool. Another thing you can do is a membrane filtration. So with this one, you'll take your liquid sample, pass it through a membrane filter, and then you'll take that filter, place it on an appropriate agar medium, and incubate. And then the number of colonies that grow on this will tell you the number of bacteria cells in the volume filtered. So if you only filtered a little bit of it, you would take note of that. Okay, most probable number. This one is confusing. I'm just going to say it that way, but it follows a statistical model. So that means that a statistician did this. Don't ask me where the most probable number index comes from. Someone who's good at math figured it out. I just follow what it says. So what you're going to do is you're going to take multiple dilutions. So you're going to make 10 milliliter dilution, 1 milliliter dilution, and 0.1 milliliter dilution from your original one. And you're going to take 5 tubes of each. So 5 of 10, 5 of 1, 5 of 0.1. And you're going to put an inverted tube that's filled with liquid. And so each of those inverted tubes will originally be filled with liquid like this one. However, if bacteria are growing, then you're going to have bubbles start to form. So this metabolic activity can be tracked by whether or not there is a gas chamber inside of the inner tube. And so if there is metabolic activity going on, then you would mark that as a positive. If there's no metabolic activity, you mark it as a negative. And so what you would do is count up how many positives there are at each factor. So at 10 milliliters, there's 4. At 1 milliliter, there was 3. And at 0.1 milliliters, there was 1. So then you would take this number and you would get the most probable number index, and there are going to be combinations of positives, and you'll just look for the correct combination. And so 4, 3, and 1 will tell us that there were most probably 33 bacteria per 100 milliliters. It's very confusing. I'm not quite sure where these numbers came from, but I'm going to trust the statistician that figured it out. Another way we can do this is by measuring the turbidity. Turbidity is a fancy word for cloudiness. So the more bacteria there is, the more turbid the solution will be. We can measure this by measuring how much light gets absorbed when you pass light through it. So first of all, when we look at our samples, we have a cloudy one, and this would indicate that there's quite a bit of bacteria in there. And we have a less cloudy one. Well, for scientists, saying more cloudy, less cloudy, not the greatest. What we want to do is get a real measurement. So what we're going to do is take a spectrophotometer, and then we're going to shine a light using the spectrophotometer, shine a light through the diluted cell suspension, or through the cell suspension, and the light detector on the other side will measure the absorbance. And so you can get a higher absorbance telling you that there's more cells in there causing the light to be absorbed or bounced off. If there's a low absorbance, that means there's less bacteria. And then the cell products, you can measure the presence of cells or by their products or metabolites. This is going to be things like pH. So if something's making like ammonia, you can measure it becomes slightly basic, or if it's producing carbon dioxide, you could see bubbles. So if you've ever seen like yeast foaming, that's not quite a bacteria, but you could see that there's carbon dioxide being produced, as well as any other gases. And that covers everything from this unit.